So it's really my pleasure to introduce and host Sofie Valk from MPI in Leipzig today. We've known each other, I think, for about 6 or 7 years, something like that. Sophie started her career also in the Netherlands, as I did, doing a master's in Amsterdam.
I started my life in the Netherlands.
As I also did. I think to the point where some of our collaborators are always confused that there are 2 people from the Netherlands that do cognitive neuroscience. I bet there are quite a few more. Then she moved to MPI in Leipzig for her PhD, with Tanja Singer and with Boris Bernhardt.
Yeah.
And we met through Boris Bernhardt, actually, in fact, several years later, towards the end of your PhD, I think, early postdoc.
Early postdoc.
After MPI, you did a stint at UDIC with Simon Eickhoff, and now you're back at MPI as a group leader with a rather large group we discovered.
But I'm a group leader in ULIC.
And you're also still a group leader in ULIC, so you never really left either place.
No, I leave, if you can.
And today we're going to be talking about deconstructing the social brain, which I'm sure lots of people will be very interested to hear about. So I'm going to hand it over to you, Sophie.
Okay, thank you, Richard, and thank you everybody for coming. So, and thank you, Cambridge, for giving me good weather and a glitter sweater. So, I think Richard thought I went to Cambridge, or no, to Trinity College Dublin, because I took 10 more minutes to do shopping. Anyway, so I will talk a little bit about the social brain, but also not really. And I will talk a bit about the dual origin, because some of you guys here really are into the dual origin. And I'm also very into it, but like for some reason my students don't like it that much. So happy to talk about it anytime, because I think it's an interesting way to understand the brain. So yeah, get me started. I can always stop if it's taking too long or is boring, or happy to answer questions ad hoc, but also at the end. So it's really all also up to you. So yeah, humans are a social species integrated in their social environment throughout their lives. So this also means that the structure and function of the brain is not something that is only projecting and generating social cognition, but we're also victims of our social surroundings and shaped by it. For example, through stress or our friendships, or like ad ultimo also by the social cultural influences that we're all in, shaping up various topics, including gender, but it can also include like the environment and the whole setting that we're just embedded in. However, the mechanisms connecting the neurobiology with our social environment remain poorly understood. And I'm a bit of a, how to say, so I think it's a round narrative, but I do stretch a bit modalities and thoughts and spaces in a vertical manner, because originally I'm from social political philosophy, and I think I'm still very interested in politics by means of newspaper, etc. But I also like complex systems, and I love the brain. So I try to bridge this all together more or less successfully. So yeah, I hope you enjoy it, because I do think that thinking about the brain in a vertical manner and embedding it in different thoughts and ideas surrounding it is the only way that we can also bridge it to bigger questions in mental health, but also in cognition, because you cannot understand the brain purely in isolation, especially if you wanted to understand cognition. So social factors are integrated in the human brain in 2 primary ways. So on the one hand, the structure or organization of the human brain facilitates social interactions by means of social cognition. Here, taking about perspective taking, but also empathy. So this is mainly done in regions that are part of association cortex. Here, denoted it with kind of ugly orange dots. On the other hand, social environment that shapes brain structure and function through experience-dependent plasticity, which is maybe also particularly sensible, or like, sensitive to these impact factors in exactly those regions that also produce social cognition because of certain factors, among others, developmental trajectories, where I also highlight this later. So I will touch both bases a little bit. Because the question is like, why and how and what does this then mean, how our brain should be connected? So this is then also a question about brain health and how the brain is functioned and organized. And I believe this has to be bridged with also concept of social health, which include both individual level factors, so how do we produce social cognition, for example, but also the social environment, thinking here also about partnerships and our living situation in the structure, but also thinking about emotional support and other factors. So it's a very, very big question, and I'm trying to break it up in different sub-questions with my students, also focusing on neuroscience alone to understand principles of brain organization, but then also all the waves and chaos that comes from it because we have friends and enemies. So maybe starting at the basis. So the social brain as a map from structure to function. So it's something that has been going on for more than 150 years, is that we're trying to map the brain into different cytoarchitectural territories, but also in microstructural markers such as von Bonin in 1950, but also connectivity regions, thinking about the Yeo-Green atlas having the 7 networks, but there's also many more of them having different numbers of networks, but also functional mappings. So my question in my postdoc, actually, and this is also a little bit how me and Richard start bridging paths or crossing paths, is to understand how is the brain structurally organized and how does this then link also to genetic factors. And here I used a more humble twin model, and I used my favorite measure of the brain, which is also a bit of a boring measure, namely structural covariance. Which means that if you take a group level data and you have, for example, the dorsal anterior insula, this region will correlate with the supramarginal gyrus across a population, but maybe not with the visual cortex, or less so with the visual cortex, because they have a shared genetic influence. And this is something that other people have seen, and they think it relates both to the ontogeny, so how these brain regions are shaped throughout development, especially early development, and also mutual tropic factors that spread through the brain because of their connectivity. So in this sample, I had twins, not these NASA twins. And I also had a second model to look at genetic organization by means of comparing humans with macaque data. And then I took a gradient approach. So I wanted to understand the large-scale axis along which structural covariance is organized. So here it means that regions with the same color have a higher genetic correlation in twins, or are structural covariant in macaques. And I found one axis that overlapped in humans and macaques, meaning that it's seen in 2 primate species, namely one going from more posterior regions of the brain in blue towards anterior regions in yellow. And here I compared both axes by mapping also the human brain to the macaque space and doing a spatial correlation of the map. A second axis was also found that explained the second to most variants, and here it was a pattern that differentiated more inferior parts of the brain to the superior parts of the brain. This again was also present in humans and macaques. So, okay, cool, colors, brains, but it has no meaning because there's no theory behind it what it could mean and how it could be interpreted. Other than that, we can say that roughly maybe this first gradient differentiates more primary areas for more like frontal or association areas, but the second gradient I wasn't quite sure about. And then I started talking with Daniel Margulis, and he said, yeah, you should check out this theory of the dual origin, which suggests that there's different waves of laminarization coming from the piriform cortex, also olfactory cortex, and the archicortex, also hippocampus. And then there's these waves of laminarization where you have a low laminarization with the origin, and it moves forward towards sensory modalities, and you have this dual stream idea. And this is something that you also know maybe from visual cognition, where you have the dorsal and ventral stream. And in general, it's like a common theme in neuroscience theorizing that you think in 2 ways. And so I did a spatial correlation also with a model where I took a distance metric from either the olfactory cortex or hippocampus, and I made them come together in the middle, and then I correlated how this map looked with this spatial distance map, assuming that this is an appropriate model for the dual origin. And indeed, both in humans and macaques, I found such a correlation from this map, but not in the other. And the second map, this may more relate to hierarchical organization, namely also this pattern where you see that regions with a similar microstructure are more connected with themselves, and then they get connected with the next level up organization, both in humans and macaques. And then the next slide gives it clearer, because this is like more of a form of hierarchical processing, going from sensory modality all upward toward heteromodal association areas and paralimbic cortex. What is interesting, and this is maybe also driven in the airplane by what Richard said, you should say a little bit more about the dual origin. And there's this great book about from Panja et al. From 2015 that I still have on my laptop, where he discusses the origin theory. And this is also something that Klaus Hilgetag has talked about in thinking about the brain, like where do you start? Like on the one hand, there's a lot of theorizing, thinking, sensory modalities, information goes to association cortices, and then blah, 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 mapped out towards motor cortex, towards action. So it's a very sensory first idea of understanding brain organization and where it starts, also thinking about its development and its change, and also ontogeny. So this has shaped certain ways of thinking, and this is also something that often come forward if you think about the functional gradient of Daniel Margulis differentiating sensory and association cortices. On the other hand, what is interesting is that the dual origin has an almost reverse model. So it starts as the most undifferentiated area. So here the example is the auditory cortex, and the quality of the figure is a bit questionable because I copy-paste this from the book. But what I'm trying to say here is that the theory starts at the not like the agranular areas. So in red is the archicortex, and in blue is the paleocortex. And then it extends, and there's this theory that there's a core, a belt, and now I cannot read and remember. So there's a core that is a succession of progressively differentiating areas that leads to the primary motor area in this case. And then surrounding this core line are the root and the belt areas that are more supportive. So this idea puts like actually this archicortex and agranular first and moves towards motor or whatever sensory modality, creating certain zones. And this helps us also to understand like how there's intralinear connection between regions and how these processes are, but also across them and also between these dual streams. So you have to imagine these streams are there, but in the end they get connected and interdigitated again. So in that sense, it also fits the previous model. Like it doesn't exclude each other. It's just that this model needs to be split in two different streams that come from the inside to the outside and like get knitted together. So I think this is like super fascinating and like understudied in some way of how this then may emerge from ontogeny. Yes, so this is also what I wanted to say, but because it's also a topic that may refers also in terms of dual architecture that we also see in cognition and in how the brain is organized in different moments. So gradients you can put also in a projection space, combining them together between the different sensory modalities. Here sensory motor regions in green and visual areas in blue, and then the red areas of association cortex, knitting them up. So this is actually a further explanation of the figure that I showed earlier. And what is so interesting is that social cognition is mainly positioned like functionally at the endpoints or the beginning points of this gradient in the association cortex. So what does this tell us about what these regions are doing and why may they be more flexible? So one idea would be to understand how a static, genetically controlled architecture may support cognition, which is more flexible. So how does the structure of the cortex support its function? So here there's two models that I wanted to compare in the second study. On the one hand, there's a structural model, so to say that the microstructural organization of a region relates to its connectivity, so something that I spoke about 2 minutes ago, that regions with the same microstructure have a lateral connection, but that you see as a function of their microstructure differentiation also feed forward and feedback connections. On the other hand, there's also the tethering hypothesis that says there's a certain uncoupling of transmodal areas from visual and primary areas to create like more higher order networks that just talk to each other in a circle. So this means there's a certain uncoupling of functional processes from these processing streams that are maybe more guided by predefined pathways. So for this, I compared brain microstructure, here probed in vivo using T1, T2 weighted imaging, and then we varied these probings along cortical depth, and I correlated on each region with each other region, creating a sort of individualized covariance, meaning that regions with a high correlation, so red here, or dark red, have a highly similar microstructure, and those that don't have it. And then I had brain functional connectivity, which is just the time series correlated. And my idea was that if a region has a similar microstructure and functional profile, the rows of these matrices would be connected, meaning that there is a functional connection that is higher in regions that are structurally similar, like this. And yes, this worked kind of, at least in the red areas, mainly also sensory motor areas again. And also, if you would look at the medial part of the brain in visual areas, but not so much in on other areas, namely also ventral attention in the blue areas, where you see actually reverse pattern, or at least a very low pattern of that microstructural similarity didn't relate to functional organization. So this pattern where my model worked actually resembled very much the sensory motor to transmodal organizational axis. And I also found when running heritability analysis that the regions that were very coupled were also more heritable than those that were not, suggesting that maybe this also has something to do either with noise, which I showed that it maybe was not the case if you look at SNR, but we can still not exclude it, or it could also relate that the functional organization was not so much anchored in what you would expect purely based on heritability. However, I also wanted to understand why do we see then a less coupling in these transmodal areas. And for this, I compared the gradients, so the principal axis of functional and structural organization, and looked at how they diverge. And what you see is actually that the blue areas in function, they detach more strongly from sensory areas relative to microstructure, whereas the green areas in microstructure, they are most differentiated from sensory areas in the green areas. So it seems to be like, if you would follow it here, so the high coupling is in regions that also show a structure organizational similarity, but then it seems to diverge again. And this pattern also, to some extent, resembled again this dual origin pattern of a split between two different processing streams, as you see here. So maybe microstructure and function are coupled along a hierarchy defined by evolution and early development. Then I also wanted to understand whether this could relate maybe to function. And indeed, you see that this unimodal transmodal axis differentiates also like more of a sensory motor attention classes in highly coupled area that also link to unimodal and secondary areas towards more transmodal areas that link to social cognition, but also emotion and pain. But what is interesting, if you look at the dual origin differentiation of the second axis, you see that this splits us more like attention and social cognition and episodic memory from emotion, pain, and also semantics. So it seems also that if you organize function along these two axes, you see that it's actually more of a, how to say, this is a German word, but a raster or a window of a two-dimensional space where you see different functions creating their place. So now I want to talk a little bit about multi-scale models and highlight social cognition from a different perspective, thinking also about the development of the brain and how these functions come about. And I think with social cognition, it's especially interesting that we have a theory of mind, and that some say, although this is also something that you could discuss, meaning like, do the kids actually understand the questionnaire and the model, and what does it mean to have a breakthrough in terms of understanding theory of mind? But like, let's ignore this a little bit and assume that there is a certain developmental breakthrough of a particular sub-function of theory of mind, meaning that kids around the age 3 to 4, they start passing theory of mind tasks. So they start to understand this concept of false belief. So to understand, to have a model of the mind of other people. And people have shown that this also links to age-related changes in the theory of mind network. However, I had a love affair with the cerebellum during the pandemic, and I was wondering, like, what is with this region? Like, it has more neurons, it is super fascinating, and I don't understand it, and I want to know more about it. And then I also started reading up about the cerebellum and its role not only in motor processing, but also in cognition. And then I started wondering, what is the cerebellum actually doing in terms of social cognition? And the interesting thing that at that point, not so much was known about the developing cerebellum in relation to cognition. So it seems that there's also protracted cerebellar developmental periods, making it also susceptible for environmental stressors, and there seems to be critical periods in its development. And then, thank God, Katharina came to my lab and she was like, yes, I want to work on this. And I was like, yes, cool. Because we wanted to understand, like, how does theory of mind emerge and how does this change the structure and function of the cerebellum? Or, this is causality wrongly put, like, how do the changes of theory of mind coincide with the maturation of cerebellar-cortical connections? So we knew already that there was maybe a link with social cognition in the cerebellum in adults, namely especially in these areas here that are across 1 and 2, here and here. And the interesting thing about the cerebellum is also that the lobules per se are not functional units. So if you do a functional map of a certain task upon the cerebellum, you don't see it linked particular to any of these levels, but it's organized a little bit differently. So that's a little bit of a side note. And there's the idea of a cerebellar generative models of how cerebellum is putting, once you create a model, it creates a certain stability of thought. So only when you need to adapt it, it gets involved. So to answer this question, we used open data from OpenNeuro with children, 41 kids, adults 56. Then we had the Partly Cloudy movie, which was annotated for theory of mind moments and pain moments, and an autoscanner false belief test. And then we compared theory of mind and pain scenes, but also cerebellar atom clusters. We did functional connectivity and affective connectivity. So first, we see that there's increased activation in social brain regions with TOM emergence. So kids with TOM, as you see here, they already have this theory of mind network. And you see also, maybe this is a good moment to use the pointer. Yeah, so you see also this region lighting up, which is not there yet with kids without TOM. And you see also that the theory of mind network in the cortex is a little bit of a mess. And in adults, you see a pattern that is quite similar to the one with the kids with TOM, but here too, but not that without. And indeed, then we also looked at the connectivity of the seeds that we found implicated in theory of mind. And you see that kids with TOM have a way mature connectivity profile of these regions involved in social cognition relative to kids without TOM. So overall, we show that there's this link between theory of mind development, where we see that overall there is this pattern of connectivity, but it gets much stronger once kids develop theory of mind. Sorry, now I had more of this. Yeah. Okay. Oh, wait, that's why I'm thinking in the wrong direction. So then we wanted to know if there's any relationship of the dynamic causal modeling, so how much the seed activation affects the target activation in the context of the experiment, and using the directionality of connections between the cerebellum and the cortex using the theory of mind clusters as a model. And what we see is that children with TOM, they have unidirectional connections from the cerebellum to the cerebral cortex. On the other hand, for adults, we see it that is the other way around, and this aligns very nicely. And although, so I don't know if any of you is the reviewer here, like we can really question DCM in the context of the cerebellar Bolt signal. So there are really methodological considerations, but we did our best to check for these notions, and overall the connectivity profile per se, not looking at effective connectivity, also aligns with this idea. But it's very interesting, and this is also the next slide, that this relates also maybe to a movement of a forward model to a inverse model of how the cerebellum is involved in social cognition between kids and adults. So this links very nicely with how the social cognition processing relates also to cortical cerebellar connectivity. So the next step would be also to think about association regions that are involved in theory of mind and social cognition, but also develop, especially in adolescence. So this is work of Valerie Sidnor, where she showed that especially regions in purple are those regions that mature a little bit later in adolescence, whereas those in yellow, the sensory areas, mature earlier. And this is also speaks again for this model where you have this sensory first idea, and then later the association regions join. And this is a pattern that you can see in cortical thinning, but also intracortical myelation, white matter development, functional refinement, pyramidal pruning, and also PV and maturation patterning. So many things happen later in adolescence, especially in these association cortices. So with Amin, and now I'm going a little bit out of the social brain towards, well, the brain itself, because we wanted to know, like, how does excitation and inhibition change in the brain during adolescence, to also understand how this may relate ultimately to individual variability. So we know that there's a big change in excitatory glutaminergic synapses, especially also in adolescence. There is like this balance between excitation and inhibition that relates to signal-to-noise ratio, but also tuning and oscillations and dynamic stability. So for this project, we used two data sets, PNC and Imagine data set. We did pre-processing, and then we did a model simulation optimization to create an EI balance in development in single individuals. And this is something that Amin was the first to try, because usually these models are applied on group level data. So this was a lot of work, and also we were sometimes a bit discouraged that these models are very hard to apply and made reliable at the individual level, and we use a lot of hours or days, months on the supercomputer to optimize it. But in the end, we fitted this model based on dynamic functional connectivity and also stable static functional connections to see to what extent the model here of excitation inhibition and based on the structure connectome can predict function. So then we set different parameters, including global coupling, but also regional excitatory weight, but also inhibitory excitatory weight to understand the different parameters. And then we did an optimization to get the best prediction again and again for many, many times. And then we create this excitatory neuron input. And then for every individual, we had a map of which we can then compute potentially changes with age. So what is nice is that we see an increase of inhibition with this model in the PNC dataset, which was cross-sectional. So this is just a similar correlation or linear model with age, where you see the blue regions show this increase in inhibition. But we see this also in a longitudinal model, so that we do a before and after model. So it's nice that in two different models, we see that our individualized approach kind of worked. However, what is maybe also a very relevant aspect that we learned of this model, that if you get a very complex models, fine deviations of your priors, they make the model also look different. For example, if you use the same structural connectome for all individuals to make a prediction, or if we say certain prior maps that we use to tune the model at the regional level, or if we take a little bit different nodes, or so we did here, and this is at the moment we're also in the review stage, so we do all kinds of models, and you see that there's a big variation, or a subtle, too big variation in each individual map, but overall, we always see this reduced inhibition in association areas. But I would say we haven't said the final word about what the best model is to map this inhibition, because both me and Amin were also like, okay, it's really, it's not an art, it's the science, but this tuning and overfitting of very complex models on data that is not perfect, based on data that is not perfect, namely the structural connectome, is a lot of work, and it's very hard to get something where we know this is a good individual marker that is also relevant for future work. But this is like an ongoing process. And yes, it's obviously, because we have this sensory association pattern, it also links to these functional processes linked with social cognition in association areas, where there are those areas that show maybe a decrease of inhibition. So red regions relate more to movement and multisensory. So then, I don't know if some of you were here when Maaike gave a talk at some point, but we also wanted to understand, like, one of the factors that impacts also inhibition and excitation models and the tuning that we see in adolescence is myelin. So glial cells are the support cells of the brain, and usually they're maybe not valued for what they are, but a specific subset of the glial cells is oligodendrocytes, namely also myelin. And myelin enhances signaling speed and efficiency, and it's also something that changes a lot depending on development, but also as a consequence of stress and other factors. So it's, I think, a very interesting measure that is highly plastic, and it increases during adolescence, and it can also attenuate plasticity. And we wanted to know to what extent does the adaptation of brain development relate to the experience you have during your childhood, especially adolescence. So whether you have an adaptive brain based on your experience versus a more susceptible brain based on your psychosocial experience. And how does this link to maturation? So as you see, this is a quite complex three-way interaction that we have envisioned. And thank God for the ANSPAN data set, because here we had a good, reliable myelin marker, but we also had behavioral information of many kids and longitudinal data to answer this question. So on the one hand, we created a prediction of how life events related to an overall distress score. So how much what you had actually experienced related to how you were feeling. And then we looked at how the predicted distress related to the true distress. And then the difference between these two factors, they relate to resilience. So if you were feeling much happier than would be expected based on the not so nice experience that you had in the past time, or the other way around. It would indicate whether you're more adaptive or more resilient to stressors. And what we find is that there's a higher rate of myelin-sensitive MT change over the course of 1 or 2 years of the study visit in those people that had a more resilient outcome. So it means that for them, their development may have gone a little bit faster to those that developed than those that didn't. Second, we also wanted to understand what is then the intrinsic functional consequence, and we used resting state data. And then we find also that there's maintained levels of functional connectivity to the default mode network in those regions that show also this accelerated change in myelin. And then we wanted to understand how does this then link to the system level maturation, to understand like how does this then relate to the reorganization of brains and the coordination of different myelin profiles and the covariance of these different organizational axes. So what you see is that there's different maturational modes. That regions that are red, those are like overall more conservative, so they change. Like if something is similar before, it becomes more similar after, whereas the blue regions, they are more disruptive. So maybe they were similar before, but after adolescence, they are a little bit more different. Hence, it creates a negative value between baseline and change. And then we wanted to see if this is then in any way different in people that develop to be more resilient versus those that don't. And we found that there's a stronger reorganization with increasingly resilient outcomes, meaning that there's maybe more of a shakeup going on in those kids that were more resilient relative to what they have experienced. And this mainly occurred in a bunch of regions, but like highlighting also again association areas here based on the default mode network, but also, well, here this is like attention network. And it was not particularly favoring one cytoarchitectonic class, although we see that it's mainly in eulaminate areas. So overall, Maike's study could show that there's a link between the microstructural refinement, the rate of myelination, and the reorganization, which all go up in people that develop to be more resilient over time, and a stabilization, is this English? Yeah, okay, of functional networks. So my interim conclusion would be that social cognition to association areas in the cerebral cortex and cerebellum changes, and it shows a change of feed-forward and feedback processing in kids and adults. In adolescents, we again observe a marked maturation of inhibitory neurons linked to areas where we also see social cognitive processing occur, but also in Maike's study, we see this also linked to resilient development related to psychosocial stressor and myelin refinement. So there's a bunch of things that connect to each other, thinking about how our brain can be social and how this may impact structure and function. So maybe this also has some translational potential to think about brain organization in a holistic way and trying to understand like what are the principles behind this co-organization that we see. And this is another study that was actually also born in the pandemic. And here we wanted to investigate transdiagnostic co-alterations in neuropsychiatric and neurodevelopmental conditions. Because we know that in mental health, I'm not a clinician, but we know that there's overlapping risk factors to get a certain mental health disorder, including also stress, and that these all have also a genetic correlation among each other, and also a diagnosis in an individual may change over their lifespan. So maybe somebody is diagnosed as depressed in their 20s, but then in their 30s and 40s, it is a different label like anxiety. So this is something that is changing intra and inter-individual, but often overlapping. And we also know that there's overlapping endophenotypes. So brain activations in different disorders have a positive or negative correlation, but at least it's often similar regions are implicated. And we know that many of these pathologies, they don't target one region in isolation, but they're more linked to network and relationships between networks. And we know that it's a pandemic and we need consortia and open science, for which we are very grateful. So for this, with Maike, again, we wanted to understand how bipolar disorder, ADHD, autism spectrum disorder, depression, obsessive compulsive disorder, and schizophrenia shared alterations across disorder. So this is based on the idea of covariance again, but this time not covariance across individuals, but covariance across different disease phenotypes, meaning, for example, that there's a certain, the impact of the prefrontal cortex is, for example, in autism, or not for example, in this data set, autism has a positive increase, whereas ADHD has a more neutral, depression has a more neutral, then obsessive compulsive has a slightly negative, bipolar has a more strongly negative, and schizophrenia has an even more strong negative impact relative to controls. And then you can have this ranked impact correlated with every other region of the brain, and you can see what regions have a similar disorder impact across disorder, creating a covariance profile. So this is exactly what we did, and we then wanted to understand like whether certain networks were particularly impacted. And again, or not again, but in these disorders we see that there's functional regions that are often implicated in how disorders are impacted together, mainly in association areas, those that also develop in adolescence mostly based on myelination, and also are again linked to social cognition, but also memory and other functions. Then we wanted to also understand how this is then organized, and we took the top 20% of nodes to understand the gradients. And what we see here is two different patterns of cross-disorder covariance. And the first pattern, again, differentiated more posterior and also temporal areas from frontal areas, whereas the second pattern related more of a differentiation between auditory sensory areas and posterior visual areas. And this first pattern in gradient 1 also related to a general structural covariance pattern that I also showed all in the beginning that was colored yellow and blue, if you remember. And this is a covariance pattern across individuals linking to this hierarchical differentiation of sensory and transmodal areas. And what is interesting is that you see that regions that are impacted together relate also to independent maturational causes and distinct vulnerability windows, maybe relating also to different functional processing levels if you bring together these different axes of how disorders are having a shared impact on brain organization, differentiating attention, perception, and cognitive control. And currently, Nan, who is doing a rotation in my lab, is also extending this model to substance use disorders because there's a big overlap between neuropsychiatric disorders and substance use, and this makes also it very challenging sometimes to find the right way to treat individuals because they're both alcoholics but also psychotic, or it's very difficult, at least in the Netherlands and in Germany too, to kind of do these people that have double hits. And at the same time, what is so interesting from a scientific point of view about substance use disorders is that it's on the one hand something that probably comes within the brain, making you more dependent on substances, but being an alcoholic, for example, itself is also not good for your brain. So it's like this two-side way of how your brain is probably altered by being dependent on a substance, but also by being a substance-dependent individual. So here I wanted also to apply this covariance approach to dive a bit deeper in it. And here we had alcohol. As you see, don't drink alcohol. It's a very blue brain. Probably also... Okay. Oh, thanks for the laugh. But no, but I was a bit like, oh, shit. But of course, it's not good. Cocaine, methamphetamine, nicotine, cannabis, and opioids. It all makes your brain a little bit thinner relative to controls. Oh, no, thicker. Sorry, the blue is positive. Is it? No, sorry. This was weirdly coded. But so it makes it thinner. And what is kind of interesting, I find, is that if you make gradients of the cross-disorder covariance, you see that if you look at the pattern that I just spoke about of Meike's study with the transdiagnostic psychiatric diseases, you see very clearly that it relates to covariance gradient 1 that was previously yellow and blue, namely that it differentiates more posterior from anterior areas here. However, with substance use disorder, this pattern doesn't exist so much. So it seems that something else is going on, and actually spatially it relates more to this dual origin in how it impacts the brain than to any other of the axes that I compared it to. And here you see that it's also a bit of, like, well, to be honest, it's a bit of a noisy signal, so I really have to figure out. But what I found initially, and it's an ongoing work, but what I found interesting is that in neuropsychiatric disorders that link also to these developmental processes, you see actually that it links also to this hierarchical gradient that I see in covariance. But for substance use and how it impacts the brain, this is not really the case, but it's morally also anchored to this more like orbitofrontal and limbic areas where it seems to be impacting it most. So then I'm also almost at the end. I think I've also talked a bit too much about too many things. But transdiagnostic patterns align with anterior-posterior patterns of cortical hierarchy and impact transmodal regions. And there seems to be a differentiation of neurodevelopmental disorders and substance use disorders, mainly also how substances impact the brain and what areas are maybe more vulnerable for neurotoxins. So it could also be a differentiation of how the symptoms are impacted. So last, I want to end with like a more hopeful, happy end. Like, how can we actually maybe also by being nicer to each other help our brain by like, and this is maybe the more fluffy form, but can we learn social skills? And if so, would this impact how our brain is organized? And this is something that also started in my PhD, and I have to say, like, this is like not it's not a meditation program per se because it was very secular, but we tried over the course of 3 months of time, first train attention mindfulness by doing like a body scan from your head to your toe or focusing on your breath. And then we had a crossover design comparing like training compassion and affect to thinking about your own emotions, thinking about your partner's emotions. So this was a dyadic exercise where you highlighted positive and negative experience of your day, of Richard's day, and then I realized, my day was also not so nice in some parts, but in other parts it was, and this is like generating emotional granularity. And the last part was also about perspective taking, so that we understand that we all have different roles. I'm a scientist, I'm also a parent. Rich and I talk a bit about being a scientist, but also about being parents, and also about managing a lab, but also about research questions. And we all have these different roles in each other. And maybe by talking about them and about recognizing them in ourselves, we can also improve our theory of mind and also impact brain regions that are implicated in it, was then ultimately the idea. And then we had another control for the first 3 months because we started with this attention mindfulness, and it was an RCT, and we wanted to know if there was also a difference in the first 3 months already. And we had a resting state control group that was just tested and scanned, but didn't do any training. And the training was like, we started off with a weekend retreat for these participants, which were like 2 cohorts of 80 individuals, where they learned the methods, and then over the course, they had like a weekly meetup, and then every day they were supposed to practice together. And actually, these people were really, really motivated because they were really happy to be part of the study. So there were actually, there was not a lot of dropout, like about 10%. And a little bit more women, early 40s. So my idea was then to relate this also to the task-based meta-analytical map, assuming that those regions that would be improved by the training would also be located in these areas as a priori hypothesis. And I looked at functional organization, namely also the eccentricity between networks, and microstructure organization, mainly measured with quantitative T1. And as you see already here is that these different networks also have a slightly different functional embedding and also a slightly varying microstructural profile if you look at the organization. There's also some water here. You want some water here? Okay. Okay, sorry. I should have. So overall, what you see is an overall microstructural change in the 3-month period, and not so much change in the retail's controls, which is good because you wouldn't expect any change. But and the negative here is a positive, meaning that there's an increase in QT1, but there's also decreases going on here in presence. And actually, part of these profiles, because these patterns are way too excessive to be real change, and it's likely also related to the test-retest effect that people just lie stiller and that there's just something that is non-neuronal going on here. So we see some effects, but they are also to some extent a little bit overshadowed by the measure that we take and the imperfection of this measure. That made it also kind of tricky to know what was signal and what was noise, even if I compared people that were as long as each other in the scanner or in the training. But what I could say is that overall, if you compare, you see patterns that are diverging, like in the retest controls, you see that not so much changed, but here in the first 3 months of the training, something did increase in terms of QT1, and it decreased again in this perspective taking. And I did this statistical test, and you see that there's also a pattern of myelination that changes, that divert between the superior and the deeper compartments, where we see that there's most differentiation in deeper cortical compartments when comparing social affective and social cognitive changes. And what's kind of nice is that it related also to intrinsic functional change, where you see that this attention mindfulness training kind of segregated the different networks, but also relates to the global workspace theory, that there's like a more relaxed network organization, whereas doing more this perspective-taking training that is more intense and you're thinking harder also relates to the more integration of these functional networks, both in attention and interoceptive networks. Interestingly, in affect, we didn't see so much going on in terms of change, may also relate to stabilization, but you can always overinterpret a no effect. So I don't know what really was going on here. And then ultimately, there was also some relationship to behavior, although this was very subtle and also very multivariate. So you do see that the model that predicted, for example, attentional change in the perspective data worked best for that particular model where you were also targeted this behavior. And same for, compassion change that improved and theory of mind change. But at the same time, I would take this with a grain of salt. And last, what I do find actually the most interesting, or one of the other interesting aspects of the study, because the whole study is of course quite nice because I did it, but I think this is kind of like that made me kind of hopeful, or not, that you see over the course of 9 months that there is this increase in myelin, especially in these association areas that are here in blue. So negative here is positive, and we saw this in both training groups independently over the course of 9 months. So maybe this is like a better time window when you can also expect more reliable plastic change in this marker, especially in the deeper areas. Or so this is something that I also find interesting. It was not the focus on the study because it looked at the specificity of each of these trainings, but you see that overall there seems to be this like increase in myelination. So then I'm also reaching the end. So I would say that it is very important to think about social brain and links between social and the brain, that we shouldn't reduce it too much. Like social is a very multifaceted construct, at least how I understand it, including cognitive and non-cognitive process. And social brain regions are impacted by various processes, including overall development. For example, with theory of mind, it's a big question whether what you see in the brain is linked to theory of mind per se, or just an aging process that is also co-occurring, and differentiating these two factors is not so easy. Like statistically, you can always do it, but really understanding what is going on is another thing. And then there's also hormones that we should consider that impact the brain over the course of our lives. So yeah, towards biosocial models of brain organization. So social functions are intrinsically embedded in human brain organization, and network organization of social functions is complex and differs from maybe sensory principles in terms of microstructure and development. And by changing the social environment, one can change the brain in specific ways, guided by its organization. And there's a couple of ingredients of my future work or current work that I'm applying. One of them is to go beyond the cortex, as I'm now doing with the cerebellum, and I'm extending this also to the thalamus, because the cerebellum thalamus cortical tract links to each other, but also diving deeper, and again in this dual origin idea, also linking this to the amygdala, which links again to the piriform cortex. And also, well, hippocampus technically is part of the cortex, but it's part of the allocortex. And there's also other work I'm currently developing, focusing more on the striatum. On the other hand, I do think one of the ingredients to understand how biosocial models and brain relate is to understand the principal axis of how the brain is organized, because this helps us to place like certain functions and theories within a broader framework of evolution and development. And the last thing, and this is like something that links also this idea of hormones and also development and rapid plasticity and adaptation to stress and other factors, is that myelin is a super interesting marker that is really like undervalued, I feel. And I feel that if all the resting state studies were myelin studies, we would maybe know a little bit more about the brain. So this is another ingredient of my current work. And yeah, what do I want to say here? Yeah, I do think this axis is kind of interesting to think about, and thinking more than one dimension about how the brain is organized and how functions relate to each other, maybe in one axis, but are separated again in another. Again, this is a very reductive model. I don't think that the 2D space can at all capture in any way functional properties, but at least it's a starting point to think about it in a multidimensional space of connecting different processes together. Yeah, so it was only made possible thanks to my awesome team. This is a bit weird photo of me, but it's the latest photo of the lab. So ignore me, and congratulate those people for doing awesome work, and also my collaborators. And Richard, thank you for inviting me, and yeah, my funders. So this is, wait, it ends here, but yeah, okay. Yeah, this is Jülich and Max Planck, so it's a bit different from Cambridge.
time for questions. Before we get into questions, I should mention we're going to the pint shop afterwards for drinks. So if people need to sneak out now and want to come back later and join the group, that's totally behind. With the second rule that have to leave now, we'll dive into any questions.
Oh, I knew that. All right, have a good weekend. Are you German or Dutch?
I'm Dutch.
Where are you from?
Amsterdam.
Oh, classic capital. Nice. See you. Bye. Yeah, so I have a question. On one of the slides, you showed like the different brain regions that like differences with various medical conditions like autism and ADHD and stuff. I'm just wondering, like in the real world, there's a lot of comorbidities. Like if you have one condition, you're actually more likely to have others. So, given the fact that some of these brain scans showed like opposite directions in honesty, how would that manifest if someone had two or more conditions at the same time?
That really depends. I think like diving deeper in this and also like work of Ashley Segal has shown that yes, you can categorize disorders and it's a very helpful framework, although there's other frameworks as HiTop and RDOC that look more at symptoms and how it impacts the brain, but overall the individual brain of a disorder is not so categorizable to certain clusters. So yes, you can clusters if you know the labels a priori, but if you just throw in these data and then do some clustering algorithm, you don't find per se these labels again back in the natural data. So that doesn't mean these labels are not true, but it does mean that you cannot really extrapolate this to any individual. You could maybe assume that somebody that has autism and ADHD and maybe also a depression, probably there's something different in their association regions, thinking that this is like a general principle that is common to all these disorders, whether what direction it goes. I wouldn't know. It depends on their age and their sex, on the scanning movement. So there's, so many dimensions there. And in that sense, I think it's also a big open question to kind of bridge from the clinic to like these maps to really understand what is going on and these multifaceted diagnoses. And maybe even if a person has these diagnoses, it may still mean that there's still a common symptomology underlying it that aligns with these different disease categories, right? So it's very, very challenging to kind of filter this out. And for Enigma, for example, they have these standardized disorder maps, but what is happening under the hood, right? The biases that each of them have, that is what we don't know, and maybe what we have to know. And maybe for this Secan Atlas cortical thickness is not the thing that I would now say, okay, we invest like 10 million in to get really this sorted, because it's maybe not where the answer lies. But maybe this is too cynic, I don't know. But
thank you.
Yes.
Any other questions?
I just asked, like, when you were analyzing those data, have you found any like significant like connections between like from the age development to like a threshold for diagnosing certain like types of disorder? If it doesn't make sense.
Yeah, I think there's overall certain disorders that are diagnosed at a certain age range more likely. Like somebody that is like 60 or 70 plus will likely not get schizophrenia diagnosed by then. Or ADHD is, although now it's changing, but it's more common to get diagnosed in childhood than in adulthood, or autism. But this is all things that are very tricky, right? Because it also has to do with the patients group that get more focal, or like with autism, you have an overdiagnosis in boys relative to girls, but now this is maybe also changed because there's a different perception of it. So I think it's, in the end, disease is not a human construct, but the labeling is. So this makes it maybe also very hard. Like I wouldn't go, and I wish I studied medicine and I was a psychiatrist, so then I knew more about it. But my partner is, and sometimes we talk about it. And what I get, like, it's also a bit, it's not so certain, right? Of course, there are very clear ways one can maybe diagnose somebody with schizophrenia based on a number of categories. But for other things, differentiating anxiety from depression, or maybe even some forms from ADHD from autism. In some individuals that are high functioning, it's very, very hard. So these categories can never do justice to the individual, but it's maybe also needed for insurance purposes to get somebody help, right? It's a system also that it's reflecting.
So well, I didn't make that clear enough because like I was curious about like when you were talking about the connectivity on the social aspects, like when you were doing some kind of like participant screening and that kind of things, like from the database, like what kind of like methods do you use like to sort of like try to distinguish those factors from the cause of like those diagnoses?
So in all the studies that I presented, I didn't, or other than the last study, what was part of my PhD with the mental training, I didn't self-recruit, but I know that usually what they do with theory of mind, that they get a bunch of kids, they pre-screen them to know that they have a good distribution of age range and theory of mind. But of course, you then have most younger kids don't have theory of mind, and most older kids do have theory of mind. So you cannot like fully have like an intermixed age range of kids between 3 and 4, and half of them has theory of mind passes or failures. Or with them, you have to oversample to get like a good distribution of that and kind of balance it out. But there's different strategies to do so. But I didn't apply them. I just wanted to have brains that have the cerebellum included, which was another thing that made it complex, because the older the kids were, the bigger their heads and the less cerebellum, and we wanted cerebellum for our study. So that was, I guess, our main criteria, which made in other ways the sample a little bit less balanced.
Really interesting, thanks. I was just thinking about one of the studies that you presented about the stress and the maturity of the myelination mechanism, and how you saw like the more mature pattern in people who were resilient.
Yeah.
That reminded me a lot of like kind of stress acceleration idea, and like that you see with functional stuff as well. And I wondered if you think that that is necessarily like a good thing, or if it might come at the cost of something later, or just what you have ideas about that.
Yes, I also read somewhere like one tricky thing with resilience is that maybe it comes back harder later. So we may see that people adapt, and here also the sample is fairly healthy, right? It's also maybe a U-shaped curve of how people adapt to stressors, right? And how, for example, war impacts your brain, and how you adapt is maybe something very different than, well, I don't mean as mean or but like a subtle form of bullying. Or I don't know, bullying is of course a real problem, right? So, but of course it's different from war. And whether adaptation to such stressors has the same mechanism, or if there's a difference in resilience to while being healthy. So it's very tricky, but one thing that is now more and more that resilience per se is not good, because it also may mean that you have a way of dealing with things that later in adulthood means you don't talk about your problem. Or I don't know, like there's a psychological component and maybe also a neurological component that if you force your brain to mature fast, then, or you also have these studies of malnutrition in Second World War, right? And that later they got cardiovascular disease more likely. So, you know, there's always time as a component, and what you do earlier in your life may backfire later, but there's too few data sets like this to investigate these questions. But yeah.
In the large data sets that you've used, there are many, many participants, but there are also very, very many brain parameters. Now, you reduce those to some higher order parameters to work with. It's the only way forward. But still, there are a very large number of those. So when you do your modeling, what principles of method do you have to guide, focus, replicate, constrain, eliminate biases from your intuitions, which you have to use to drive to get the whole thing started? Can't get away from that. It's a sincere question. I'm not trying to trick you, but what principles of constraint do you, of methodological constraint, which are not inside the stats, inside the modeling package, that you apply to diminish problems of bias and arbitrariness in such modeling?
I think ultimately one cannot fully like remove all, but
sure.
But I think it depends really on the study of what we do. So one of the methods is that we use multimodal approaches, so that we see something, for example, in microstructure, and we try to link this to function, knowing that these are different imaging modalities. And if we see something more or less consistent in two modalities, it's in a way...
And how do you quantify, that's good, it's sort of a meta-replication, if you like. How do you quantify consistency when you have quite abstract parameters in each domain?
For example, like in Maike's study, we see frontal cortex changes, and we see that this links to functional connectivity change, and this does more so as any other region in the brain. So it seems to be specific. Or in case of Amin's study, we did a lot of novel modeling testing and a lot of statistics, and we also had an external replication data set that used a longitudinal design next to the cross-sectional design, and we looked for convergence here. Or in Katharina's study, it is very tricky because it's only too few kids, right? But there we used, and also this is something that we often do, that we use different methods and we see if at a conceptual level they tell the same story. So in the case of the DCM and the local changes,
Sure.
So it really depends.
Somewhere in the conceptual levels is a good way.
Yeah, but you're completely right. Like with such diverse setups of different types of studies, they also need a different way of control. Like some of our studies we do already pre-registration, but other studies we use different either statistical methods or use another data set if possible, or use a second modality if possible. And I think being skeptic and training my students to be skeptic of too good results is also another metric. And at the same time, many of the things I've told, the individual result will likely not hold for the next 100 years, right? Like that would not be my legacy, this one region that shows a change. So I try also not to overinterpret this one dorsolateral area that I saw significant at a certain threshold in my underpowered study in study X. That is not my heaven. So yeah, but it's hard. I agree.
I know.
Yeah.
Thanks, Sophie, for a great talk. And you've really produced an astonishing amount of work in this area. I just wanted to follow up on Mark's questions from a slightly different angle about, where do you find an anchor point in this kind of like universe of imaging phenotypes? And one strategy that I've kind of found quite powerful coming to it late in life is genetics. But I don't know how well that would work for social cognition. Other GWAS scores that you would believe as kind of anchor points for thinking about the origins of social cognition? Obviously, if you have a GWAS on social cognition, you trust you to begin to explore and de-randomization type approaches, I imagine.
Well,
Identify brain regions that are causally related to that, for example.
I think, yeah, well, bin one is with Richard and Varun also working on GWAS of a gradient organization. So we do branch a little bit out in genetics, and I've also done more complex social equation modeling in twins. Yes and no. I think genetics come with their own caveats, and I think certain phenotypes can actually, like, genetics can maybe say something about social cognition or the regions that help these processes, and maybe your susceptibility to stress, or maybe per se, if you think about, for example, sex differences and how this impacts certain, so down the line, yes. However, currently I'm also really liking also animal models and extending this not only in human species, but in other species to see how my model holds if I, for example, and this is like not funded at the moment, but my neighbor has zebrafish, and she's my friend, and she wants to write a grant on like zebrafish and stress models and ADHD, which is in itself something you can question, because how do you know if a zebrafish has ADHD? But I do think that if you create like these vertical models, as you also do with genetics and network organization, but also with different species, it's one way forward. But I don't think there's certainty at any level. And this is why I try to find some consistency at many levels. But I would also say it's, at the moment, as you say, I've been very productive, but I think it's also now a time to think. And I do a lot of thinking, but maybe not enough to I would love now to find time to write a review and to really collect these ideas that in my head are all connected. But I also see in many of my trainees, they are like, okay, I want to work with you, but it seems you do everything. And to me, that's not the case. There's many things I don't like and don't do and know nothing about. But to make this into a coherent story, I think a review would be something that I would think that collecting the evidence and, yeah, would I advise to myself. But then there's another project that I like doing. So, but yeah, no, you're completely right. Genetics is one way of going.
Yes, I'm asking to follow up on the cross disorder covariance gradient, spatial gradients research, because it's really interesting. So, do you think there will be a Langton's principal dimensions that's going to describe general mental health that can be inferred from brain imaging signatures? And how do you anchor that to the social and environmental, for example, social skills, and that sort of phenotypes that can be derived in a more social context?
I think there's a lot of work on the P factor, like a general factor of mental health. It's hard to say. Some people have placed the P factor in the cerebellum, which would be a great coincidence of my interest, but it's how you define it, and also how, like, I also was at a place where I had to do a thesis, and then we discussed also, like, can you actually equate symptom variation in a healthy group of people with diseases, or is there even a boundary of healthy and disease, or is mental health a continuum if you think about symptom dimensions? And I think there's not enough knowledge at the moment to decide that you are healthy and I'm sick based on a certain skill, right? In the end, it's also about, like, perceived burden of you, your family, and these definitions are all pretty cloudy, and they cannot reduce to the brain itself, right? Like, if my family is suffering and I'm perfectly fine, and my brain is, like, at least in cortical thickness, looks quite normal, like, it can still mean you're sick at some level, right? But it may not be so easy. So I don't know, like, in the end, I think time and money needs to be invested in an individual, and not only reducing it to the biology, but also seeing it in the context that this person lost their dog, and it meant everything to them, and they're more susceptible to depression, and now they don't go outside at all, and henceforth the whole cascade. But this is maybe more meaningful than the superior temporal gyrus being a little bit thicker. Like, what does it then mean? Like, maybe for me, my brain is also sick, but here I'm standing giving a lecture. Like, you know, it's also a context that matters so much. So I would say that maybe, sorry, I give too long answers, but one thing that is lacking in the current databases of diseased patients that are shared is enough information about these individuals, because often you just know the label, you don't know the comorbidity, you don't know if they have a cat, you know, but these things do matter if you're a psychiatrist treating a child or an adult with mental health conditions. Like, what is the family context? Like, that's, like, not that I'm all going, like, social psychiatry, but this dimension is, of course, there, and it's, like, now unable to study, right? Because we don't have that information at the scale that we would need it to decipher the complexity of the system. Yeah.
Could you expand a bit on the cerebellum differences that you saw between children and adults, and the forward model and the sort of feedback model, and what do you think that means?
It could have to do with automation. So that first, with kids, it's still a challenging task to do theory of mind, and it's not yet automated, but it has to be guided by the processes that are supported by the cerebellum before the processing takes place in the cortex. But then, when you're an adult, you're also more experienced mind reader, so to say. So for this, you don't need the cerebellum anymore, because the cerebellum only starts to interfere in movement if you want to change a movement, right? But maybe, similar with theory of mind, you don't need this interference anymore if you're an adult.
So you rely on priors?
Yeah, at least in the tasks that were measured there. But if you have a very complex theory of mind task, you would still need the cerebellum to adapt and adjust your precise model that you have. So, but in the context of our study and how the people were measuring theory of mind with the partly cloudy movie and false belief test, we see this. But then what it truly and really means that comes with other challenges, especially like forward feedback model. It's all beautiful and nice, but the DCM model is not perfect because the bold signal in the cerebellum doesn't link to vascular activity in the way that it does in the cortex, which is already a big limitation. But per se, theory-wise, the idea makes sense of the role of the cerebellum in relation to the cortex. It's just that we need more data, better models to then test it.
I know lots of your research are going from the aspects of like imaging and sort of like data analysis. Like, do you have any sort of like tasks-based evidence on like the judgment of like whether someone has developed a theory of mind?
Yeah, in this case it was based on tasks. So you have like a false belief task where you have this Sally Ann paradigm, and there's this cake, and then the mother is like, oh, this is a cake. I will put a bucket over it, and another bucket I will also put next to it, and then the child watches, the mother walks away, and then it's suddenly shifted. And then the question is, does the mother know where the cake is? And then the answer should be no, of course not, because the mother wasn't there when she saw that the buckets were changed. And the kids, if it has theory of mind, knows that I don't know what I don't know. But if it doesn't have theory of mind, it assumes that I have the same knowledge as they have. So as if I would have been, so their model of their mind is their model of my mind. So this is one of the tasks that you could apply, for example. But this is, for example, in adults, much more complex, because we all know that the bucket has moved, right? So to understand differences in theory of mind, like some people are really better than others, I would say from experience. But, you know, to test this is another thing. And then it becomes very linguistics and meta models of, you think that I thought that you were asking this question after this and this, that happened. And then it comes like, okay, yeah, maybe you are not able to do recursive thinking or whatever. So then it gets also related to other factors. Yeah.
You might give an example of like the thing, one of the tasks like you use for, or like the data. Yeah.
Well, so this thing I said, that is commonly done in kids. And in adults, you could, for example, have like a story that I would tell about, I don't know, I would say, oh, my brother died. I bought like this flower setting. And then, okay, what is now the theory of mind story? The flowers was upset that I wanted to have red flowers because I originally ordered black, but then I changed my mind. And then there is like an ABCD question that says, Sophie thinks the flower person is upset, but in truth they are not, because they were just stressed. Or Sophie is just losing her mind because she just lost her brother, so we cannot trust her story. Or something else in relation to the story. And then there's one true answer relating to theory of mind abilities of you about my current state of affairs. And you would contrast this with a factual question like, oh, the flowers were black and then they had to be red, or there were no flowers but there was a florist, or I don't know, like some factual, so that you understand the story, and then you compare how well people are understanding the story versus how well they make inferences about how I was thinking in that situation.
So is this kind of task like embedded like before the testing and collection of the data?